This is a video short that goes with DNA Structure and Replication PowerPoint that is also located there on the website. These are meant to be like short videos that cover about 10 minutes of topic, and you can go through it, stop it, rewind it many times you want. I would advise making some flashcards that go with this, so that when you get to something that you know that is important, you could get an index card and write down flashcard, maybe a structure or a question on the other side, the answer. So here we go. First of all, DNA structure. We're going to talk about DNA structure, and here's a student model, one that students made, that is a really good model that has... Looks like we lost our video here. Got the timeout set too short. So this is a really good structure that student made, student model. You can see it's in the DNA helix. This shape is called a double helix. That's an important concept, that you know that the shape is a double helix. Why? Because it's like a ladder. It's got two sides, and it's a helix. A helix is a spiral. On the outside, the rail of the ladder, it's a twisted ladder, is phosphate, sugar, phosphate, sugar, phosphate, sugar. The sugars are where the DNA, or where the nitrogen bases are. And in the middle here, like the pink and the purple, these are the nitrogen bases. The letters, G, T, C, and A. And always G pairs with C, two round letters go together, and A with T, the two letters that have straight sides. So A and T, G and C. On the rails, though, it's a repeating structure of sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, phosphate, and they're identical all the way down. And you can see again that this just a long helix shape, and it's a very, very long structure. This only is a very short segment of it, and these DNA, or this DNA structure, is what makes up the chromosomes, and it's where our genetic information is kept. The nitrogen bases, the letters, the order of the bases, store that information. So, move on. DNA occurs in a twisted ladder shape, makes up the chromosomes in our nucleus. We have 23 pairs of chromosomes. The rungs of the ladder are made of phosphate, sugar, phosphate, sugar, phosphate, sugar, identical all the way around. And of course the nitrogen bases, A always pairs with T and G always pairs with C in DNA. That's the structure. In between the bases is a hydrogen bond, and it's a weak bond. It's kind of like Velcro. So this molecule can unzip and zip back, either like Velcro or like a zipper. It can actually come apart and put back together again. And that's handy because when it replicates, it has to come apart in order to duplicate the strands. And this shows you a little bit about how this just comes apart like this very easily. And where there is an A, the new strand is made a complement of T, and where there's a T, the new strand in A is made, and once it's through with all that, it has two identical strands. This replication occurs during interphase, so we can kind of tie this together with mitosis and meiosis. Interphase is when this duplication of DNA or replication occurs. So here's a picture of the DNA structure. If you've downloaded this PowerPoint, which I advise you do before you go through this video, you can see the way it looks when it's spread out, the way it looks when it's in its helix shape. This is more of a 3D model of what it really looks like if we were able to zoom in with a camera all the way down to the size of DNA. So we're going to move on down and talk about replication. Replication is copying of a DNA strand into two identical DNA strands. And other than maybe an occasional mutation that takes place, the two strands are identical. And that means that our chromosomes, now instead of 23 pairs, we have two sets of 23 pairs. So that when it splits into two cells, we have a set for each cell. And that's the end result of mitosis, is two identical cells in mitosis that have the same DNA structure other than maybe a few mutations that occur. So this is important. Replication is important, and it occurs, again, when this unzips, and on this side, a copy is made complementary. On that side, it's complementary. You end up with two identical strands. So we move down a little bit, and you can actually see the process of replication. Here it's unzipped, and the polymerase comes in, and it actually grabs a T and puts it on the A, and grabs an A and puts it on the T, grabs a C and puts it on the G, and you can see we're starting to get a new strand here and a new strand on the other side, and the end result is this separates and you have two identical strands, which results in the end in two identical chromosomes. So that's the basics. Like I said, I would go through this again and make some flashcards, get some index cards. I've got plenty of these down in E106, and there will also be some in the bio room. Get some index cards, write a question on one side, answer on the other, and you can make a deck of those that you could look over as you study for the tests for this unit. So there are other sorts. This is one of them. There are other sort videos. Pick a topic, go through, and each of them are somewhere between 5 and 10 minutes. And that will be short enough to where you could focus and get some good information. Come down to E106 or come in your bio room if you need and get some help. Mega lunch in mornings. Good luck.